So I'm Emily LaPlante. I'm currently a PhD student at Baylor College of Medicine, but I also act as the data coordinator for the Xrna Atlas. And I'm excited to share with you today how we're planning to look at the Xrna Atlas from the RNA binding protein perspective. So since this is a workshop, we kind of wanted to make you all excited and thinking about some of these questions in the field of RNA binding proteins, specifically in the context of extracellular RNA. So obviously, I'm not going to answer all of these questions. The earlier talks haven't answered all of these questions, but just to get you thinking about it and a bit excited. So one beginning broad question is simply, if all RBPs are released into biofluid, how many RBPs are released into biofluid of those we have characterized? Are there characteristics of these released RBPs that might give us some information as to why or how this is happening? What percent of RBP bound sequences are exported from the cell as exRNA? So these RBPs can bind up to thousands of sites, and some of them might export all of the sites that they're binding, some of them might export half, and some might be very selective about which ones are being exported. And so we want to explore these classes separately to start thinking about how this biology might be affecting xRNA. Are these RBP binding sequences biofluid specific, and are some of them present across all biofluids? So this is a bit of a quality control question. Is there an RBP or some sort of binding site from a given RBP that you should expect to find in your given biofluid? If it isn't there, is it a problem? The same sort of thing across all biofluids. If something isn't present, is it something that you should be worried about? But also thinking about specificity, is this maybe due to a cell type of origin if an RBP is biofluid specific versus maybe a more general cell mechanism if it's found across all biofluids? Two of the questions that I'll be touching on today are actually, where do released RBPs bind across the transcriptome, and are there high occupancy sites that are released? These sites could be difficult to interpret because lots of RBPs could be binding there. So pointing to a specific one might be difficult. But the idea that these regions might be especially important because we're seeing lots of RBPs binding are interesting. And finally, do RBPs levels change across physiological states, and or does site occupancy change? This is getting at the idea of, are we releasing more or less RBPs regardless of which sites, or are we simply changing which sites are released during disease or physiological state? Most likely it's a combination of the two, but putting these RBPs and xRNA in context of a physiological state or disease is the end goal here. So to do this, we will be using the ENCODE eClip data, which we've heard about in the previous two talks. Briefly, this data is generated when an RBP is in essence cross-linked to RNA, which is then degraded. And when you sequence, all you have left are those fragments which were protected by this given RBP. There are currently 150 uniquely available eClip data sets, and we're very happy to hear from Eric's talk that hopefully more are coming soon. Each of these RBPs has 2 technical replicates per cell type. So as was also mentioned, some of these were profiled in 1 cell type and some were profiled in 2. So there's anywhere from 2 to 4 technical replicates for a given RBP. Also, as mentioned, there are a lot of different file types available for a given RBP. You can start from FASTQ if you'd like to do the alignments yourself. You have BAM files, which are pre-aligned, bed narrow-peak files, which I'll be talking about later, and big-wig files, which are often used for visualization. So just a brief overview of what we're going to be doing. We're going to be taking our FASTQ files, which we've aligned, converting them into a format which we can then intersect with bed files, which we've received from ENCODE, and then doing some basic comparisons. So we're going to be using these bed narrow-peak files from ENCODE. What exactly are these? They're a fairly typical bed format, in this case a 6+4, where each row of this file is going to be a given binding site for an RBP. The first 3 columns, as with any bed file, are very important. They tell you where a given feature, in this case a binding site, is located. We know the chromosome start and stop for a given site. There's some other columns that you might be interested in. These can be used for filtering. Strand will tell you if it's on a given strand, if you're interested in strand-specific sites. Signal and p-value are both related to the peak collars that are used in the ENCODE pipeline, which give you an idea of enrichment of signal. So you can use these columns to filter for how enriched you want your site to be in order to actually consider it a binding site. In addition, ENCODE does already have pre-merged technical replicates where they've done some of this filtering for reproducible peaks, and these are the files that we'll be using going forward. So now that we have these files, we can do some very simple visualizations to see where RBPs bind across the genome or the transcriptome and where those transcripts are coming from. In this case, we have our two cell lines, and our y-axis is showing the number of RBPs that touch a given region, and I'm just displaying chromosome 1 here along the y-axis. We can see that our two cell lines, while different, do tend to have similar profiles of where many RBP are binding and where very few are binding. But this isn't exactly what we want to know. We want to know which of these regions are also present in human or in extracellular RNA or the extracellular space. So where do these bind that are released? And then so in order to do that, we have to actually know which regions are released. And so we turn to the Xrna Atlas, which hosts over 4,000 small RNA-seq samples across multiple conditions and biofluids. We'll be adding 3,000 samples in the next year, with more expected to follow in the years coming up after that. And so in this case, if we're just interested in which regions are present in extracellular RNA, we're not going to be filtering based on condition or biofluid, but that is definitely available if you are interested in specifics. So just brief overview, kind of visually, what we're interested in doing is using these reads that are present in biofluid or from cell lines or mouse biofluid, this case just a human, and looking at which regions are present and intersect with a given RBP. So what we want to do is look only at regions that are present both in biofluids and for an RBP of interest. So in order to do this intersect, you have to have a couple files. The first, you need to know where the RBPs bind, and this is captured in those ENCODE or ENCORE bed narrow peak files. And you also need to know where the reads align in your biofluid sample of interest. These are captured in BAM files, so these aligned files from a FASTQ file. In our case, we're using the xRNA pipeline since it's been made for small RNA-seq. However, you can't do a direct intersect between a BAM and a bed file, so there has to be an intermediary file. And in addition, BAM files are prohibitively large. And since we're planning on doing an analysis of 7,000 different samples, keeping 7,000 different BAM files is pretty difficult and space prohibitive. So what we do instead is we take our BAM files and we convert them to a coverage format, which is basically a bed format. We have our first three columns, which tell us where a given feature, in this case, what coverage is at that given location, and then the coverage value. And this is base pair level. However, if two base pairs next to each other have the same coverage, then it's collapsed into a single line. So here we're really trying to think about how to keep these files as small as possible, just so that as we expand the numbers that we're interested in, it's all feasible. So now that we have these sample coverage files, we can look at which file regions are present also in an RBP binding site from those ENCODE bed files. And what our output file will be is, in essence, the regions that intersect, that are present in both. I'm going to go very briefly over a case study here where we looked at some changes in plasma and xRNA during exercise. This study has been used before. So we took, and is present in the xRNA Atlas, they took 26 samples, male and female, and drew plasma and did xRNA isolation, both at baseline and after exercise. So we have these paired files. And what we can do is test for differential binding for all of our RBPs between baseline and peak exercise subjects. We do this using a Poisson test because we do not assume a normal distribution. And this is basically asking the question, is there differential release between the RBPs, a given RBP? So we're not thinking about specific regions, we just want to know the level that an RBP binds in pre or post exercise. In this case, it's a little difficult to tell because of the outlier in the baseline, but these are significantly different. We see significantly less release or less binding of IGF-2BP1 post-exercise at peak compared to baseline. I'm going to go over two top hits. One is this IGF-2BP1, which has been shown to function in regulating loading of extracellular vesicles, as well as the vesicles' effect on metastasis and melanoma. KHSRP is our other top hit, which is a splicing protein that is involved in spinal muscular atrophy, so we believe that it has function in muscles, which would make sense since this is an exercise phenotype, but has not been shown to be involved in EVs or extracellular RNA at this point. The next thing that we can do is look at individual sites. So are there individual transcripts or regions that might be affected? In this case, we're looking and we see that this site actually has significantly more binding post-exercise or at peak exercise compared to baseline. So even though a given RBP might decrease the level of binding at the global level, we can see specific sites that might actually increase. In this case, we have one hit for IGF-2BP1, and it actually overlaps with an exon of this mRNA ST3-Gal1. ST3-Gal1 has been found in exosomes previously, and it is known to be upregulated during exercise. So this is good that we've seen kind of this biological signal. As for KHSRP, we actually have two hits. One overlaps with an intron of BUB1. This makes sense because it's a splicing protein, so it makes sense that we'd see this hit in an intron. We know that BUB1 has been found to play a role in endocytosis, and this might be the first sign that KHSRP is involved in xRNA communication. Also, we identify a binding site in this uncharacterized link RNA, which really we know nothing about other than the fact that it's found at very low expression in the nerves and testes. It hasn't really been studied. So these are the regions, the proteins, the RBPs that we would be interested in looking more at if we were interested in studying this exercise phenotype. So I'm going to give you a very brief overview and review. So we've taken our FASTQ files, we've converted and aligned them to BAM files. We went from BAM file to coverage files, which we could intersect with bed files. We then have done some preliminary comparisons. We've used a few tools, excerpt, pretty much all the middle steps can be done using bed tools. And finally, you can really analyze your data however you'd like. We tend to use R, but there's a lot of software out there. So as for availability, not all of the FASTQ files are present on the Atlas, and not all the samples on the Atlas have FASTQ files associated with them. Because of some patient privacy issues, not all of them are consented for that. You can find them on dbGaP, but that can be a bit of a process. So starting from scratch, probably not going to be able to reanalyze the entire Atlas yourself. However, we are currently making all samples have this coverage file available. So we're going from FASTQ to coverage file, and coverage files will be available for all samples on the Atlas. In addition, we will be intersecting all of these coverage files with the ENCODE bed files, and these intersect files will also be available on the Atlas. None of this is complete yet, but it is coming. So if you're interested, please be on the lookout for it, and it will be there eventually. With that, I will thank my lab, the ERCC, DMRR, and our two excellent engineers who have helped me with all of this reprocessing, Andrew Jackson and David Chen. With that, I don't know if we have time for questions, but that is it.